Now imagine, if you will, a 6-foot strand of DNA. And though that strand of DNA is made up of nothing but these nucleotide bases. There's no spaces, there's no punctuations, there's nothing to indicate how we're going to read those nucleotide bases. And so we can end up with kind of a problem there, where what happens is, let me kind of illustrate this for you with just some letters. We're going to assume, I'll just write one strand. Let's assume the template strand of DNA reads A, T, G, C, C, G, A, T, T, C, A, G, T, something like that. Okay? If we start here to make a messenger RNA, the messenger RNA is going to be complementary to that template strand. And so we're going to get a messenger RNA that reads U, A, C, G, G, C, U, A, G, U, C, A. Okay? And so then if we look at so if this is my triplet, this would be my codon. And we can actually go back up here. No, that's not going to be useful for us. Let's go to the codon chart. I'll explain this a little bit more. We can look at these codon charts and we can figure out UAC, what that is going to give me. So let's find UAC, and UAC is right here. And so that's going to give me a protein with a tyrosine, with the amino acid tyrosine. And GGC will give me another amino acid, and so forth. But what if this is not the starting point? What if this is just some non-coding DNA and the starting point for the gene actually is right here? Okay. Well, that creates a bit of a problem, right? If we start where we're not supposed to start, we're going to get one type of protein. But if we actually start here, here's my codon. That's my first amino acid. So AAG, if we were to go down here and take a look, my very first amino acid would actually be AAG. And where's my AAG? There we go. It's going to be lysine. Totally different amino acid, totally different properties, and the protein would function completely different. And so we have this problem in that we have what we call a reading frame, which essentially means, where do we start the transcription process? Where do we start this process? And so to determine what my reading frame, what the correct starting point is, we need to look at our gene and the layout of the gene and how we would find it in this big long mess of non-coding information. So if we're going to look at that and we're looking at DNA and my gene, what we would see is, I'm going to use gray for my non-coding region, and I'm just going to draw the template strand. Okay, and I'm not going to give letters to it. You can think in your head and know that we would essentially have these letters to it, these nitrogenous bases. But in this case, we're just going to use a template strand, and this gray region would be my non-coding region. And then what I'm going to draw here is a section that's green. Okay, and we're actually going to I'm going to come back to what this green section is. We're going to have some more gray and maybe another green and maybe more gray. And then we're going to get to a region, and I'll draw that one in pink. Okay, right here. Okay, and only after that will we get to our coding DNA. Kind of ran out of a little room, but we're going to go ahead and make this thicker. There we go. We're going to go ahead and keep with this in mind, and I'll just kind of twist it around because of course DNA is very flexible. And some of this gene actually will have my non-coding segments in it, and then we'll go back to having a coding segment and maybe another non-coding segment. And then what I actually have, so we'll have a coding segment again, is I actually have another region, and we'll put that one in purple, I guess. Okay. So here's my color-coded pieces of DNA, and then we're going to go right back. We're going to have this big old long strand, and so we've got more of the gray region, like so. So let's label these. These regions right here are going to be what we call regulatory regions. I'm going to come back and explain this, what they are, but as their name suggests, they're going to regulate the process of translation. So gene translation, correction, gene transcription. This region right here is what's going to be called a promoter. And the promoter is the start signal. Going back up here, the promoter would tell me that I need to start right here. It is the start signal. It lets me know where the gene actually begins. And then my blue portion would be the first segment of the gene, and because it is coding, we're going to call it an exon. And let's go ahead and label it. We're going to have it exon 1. And then this segment right here is in gray, and we're going to call this intron 1. Here's another exon. This one's exon 2. Here's an intron. And again, now we've got exon 3. This purple area is called the terminator sequence. And that tells us where the gene stops. And so in order to go through the transcription process where we make a messenger RNA, the proteins involved in that process will scan the entire DNA strand until it finds that promoter region, at which point it's going to set down and begin the transcription process. So this is a little bit of a more complicated drawing of what a gene looks like. Here's a very simplified process. Notice this one doesn't include the regulatory regions, but I will come back to this idea of regulatory regions. It also doesn't show the presence of introns and exons in the coding region, but it's the same basic idea. You can see my promoter, which is my start. Here's where we're going to start, and my termination sequence, or terminator, which is where we're going to stop. Okay? And that's the basic idea. That's how we pick out of all of these nitrogenous bases, we can pick out which ones actually code for a protein versus which ones are non-coding. Okay. Now, the process of transcription translation, the process of protein synthesis, we have what we call a central dogma. Okay, a central dogma. And the central dogma simply states that if we want to make a protein, we have to start with the DNA, and from my DNA, we're going to have my template strand, or my coding strand. We're going to produce my messenger RNA. And that process is called transcription. Then from there, we're going to take the messenger RNA, which will then become the new template. And it becomes the template for protein synthesis. And that process is called translation. So transcription and translation go together, and they're both part of protein synthesis. You can't have one without the other.